Okay, well, welcome everybody. This is the second session of the 2022 autumn series, which has a theme: quantum mitochondria, energy, information, and implications for health and disease. And today we're going to welcome our speakers, Professor Karl Morten, who will be introduced by Rees Mould. So if I just introduce Rees, first of all. Rees is a postdoctoral research fellow at the University of Westminster and works closely with one of our advisors, Jimmy Bell, and has been undertaking Guy Foundation biophoton experiments, Guy Foundation funded biophoton experiments. And we're actually really delighted that he succeeded in having a fairly exciting paper on the non-chemical signaling published in Results in Chemistry last week. And I'm sure, Rees, you can probably share your link. I think that's probably available if you share your link to on the chat line during the course of the meeting. I think some of the participants might like to pick that up. So without further ado, Rees, can I hand over to you to introduce our speaker today, Karl Morten?
Thank you. Okay, as Jackie mentioned, my name is Rees, and I'm a postdoc at the University of Westminster. And it's my pleasure to introduce today's speaker, Professor Karl Morten, and his presentation, Mitochondrial Dysfunction, Relevance for Chronic Fatigue Syndrome and Long COVID. Karl started his PhD in Bath/Bath, then took under postdoctoral positions at Bristol, Oxford, and the Buck Institute on Research for Aging, and now resides as the Director of Grad Studies and PI at Nuffield Department of Women's and Reproductive Health at John Radcliffe Hospital in Oxford. I've actually known of Karl for a long time as the founder and organizer of the Mitochondria conference, which is a annual one-day conference all on mitochondria held in Oxford. And it remains the single best catered conference I've ever been to. Including this wonderful showstopper mitochondria cake. I've also heard there was presentations and talks at the conference. The cake actually leads on nicely to Karl's research areas under mitochondrial health and disease, in particular, looking at targeting energy metabolism as a therapeutic strategy in cancer, identifying fatigue-associated blood factors in ME and chronic fatigue syndrome, and the development of Raman's spectroscopy as a tool to study energy metabolism. And in today's talk, Karl is going to be talking about mitochondrial dysfunction and how it manifests, and how dysfunction of mitochondria manifests in chronic disease. And such chronic diseases such as ME, chronic fatigue syndrome, and COVID remain poorly understood and under-researched. And considering our these research areas in this talk and our focus on mitochondrial health and disease in this autumn Guy Foundation series, it's hard to imagine a more suited presenter than Professor Morten. So without further delay, I'd like to hand over to Karl for what promises to be a very fascinating talk.
So thank you very much for the opportunity to talk today. It's a real honor to give a talk at this sort of symposium. For the last year, I suppose I've listened to large numbers of fantastic talks on this platform. So it's a little bit scary as well. And when you see all these people come in from all over the world, you sit there quite calm and then you get perhaps a little bit nervous, but hopefully you'll enjoy what I've got to say. And so when I listen to the talks around the quantum biology and the sort of more sort of fundamental physical areas of science, I get quite interested in how can I apply that to my work? I'm very much a biologist. I work on cell models. And so I'm always thinking about how can I apply some of the things I hear about in this seminar series to my research. And maybe today you might have some ideas as how you can apply your things to my work, and that could be quite an interesting connection. We've just applied for a rather large grant to the BBSRC for a big project looking at some of the areas I'm going to discuss with you today, which could be quite fundamental in our understanding of what mitochondria do. So hopefully we'll get some thoughts to that. And if we are successful with the grant, maybe we could think about bringing in some quantum work in that. So I'm, as Rees said, I'm based at the Women's Department at the University of Oxford in this 1970s building you can see on the left. I'll just go onto the laser pointer and make it easy to see. So old in terms of science terms, but not as old as some of the parts of Oxford. And so I've studied mitochondria and worked on their biology in health and disease for over 30 years. And we got interested in chronic fatigue syndrome about 6 years ago. This chap over here, Norman Booth, was a physics professor from Oxford. His wife had the illness, and he basically went to a meeting in Germany. He was interested in mitochondria, published some nice papers exploring this, and said to them, "Do you know anybody that knows anything about mitochondria?" And they said, "Well, this guy Karl Morten, and he actually lives just around the corner from you." So Norman and I got chatting. It did take me a while to get really involved because there's a lot of stigma in this illness and complexity into how it works. And so I'm going to talk about some of our work in that area today and cover that. And Charles Shepherd, who also is on this slide, I'm just going to remove the images here, from the ME Association, has basically funded all of our research to date. They kept us going for the last 5 years, and we're just about to start to publish some nice work. And so this is my group at the moment. So we're very interested into developing new assays. And so we have nice assays involving plate reader format. We're measuring oxygen consumption. We use Metabolomics and sort of chemistry, and work with the chemists, and some pathway analysis here by Jamie, also in my group. And as Reece said, I will talk about some of the new work we've been doing using Raman spectroscopy as a potential way of looking at mitochondria and mitochondrial function in isolated mitochondria, but also maybe cells and tissues. And so this is what I'm going to talk about today. So I know we've got quite a mixture of people on the call. So I'm going to give you a bit of a sort of a background, really. What are the challenges of understanding a chronic disease? We probably know many examples of chronic diseases. And in particular, how does an energy metabolism dysfunction in ME, CFS, and maybe long COVID fit in? What do we know about those two areas? And we know mitochondria make ATP, but what else do they do? And I'll talk a bit about their role in dealing with pathogens and how this may have an impact on some of their other mitochondrial functions, where you've got many aspects of mitochondria going on at the same time. And this, fourthly, an interesting concept, which is the basis of our big grant application, is can we make aerobic ATP outside of mitochondria in mammalian systems? And if we can, how might this be relevant to disease in general? And basically, to touch on at the end, there is quite an interesting area in chronic disease where the emergence of pathogens around the microbiome. And I will talk about the microbiome during my talk, not just the gut, but the whole body microbiome, and how we're developing new ways to try and study not only mitochondria, but also maybe these pathogens. So that's a summary. It's quite a lengthy piece of work, and hopefully you'll find it interesting. So this is a summary slide of the areas that we're interested in. So the ones in the red boxes are areas that we're actively studying. We've worked on ME/CFS for some time. Long COVID is a new project we're just about to get going on, where we're going to recruit patients to studies. Lyme disease is an area we haven't worked, but we are just about to start to. And these other areas over here, the fibromyalgia, the multiple sclerosis, the PANS, PANDAS, and the endometriosis, are all chronic conditions where we really don't understand what the underlying cause is. And they will come into the talk a little bit as we move forwards. And so I'll put this slide in just to maybe we should think about chronic disease as accelerated forms of aging in different parts of the body. As we age, things go wrong over time and things decline, but maybe these chronic diseases are an example of an aging process that's maybe accelerated for some reason in certain tissues in certain individuals at a particular time. So I said I'll mention the microbiome, and many of you are probably aware that there's more bacteria in our bodies than human cells, and we are mainly microbes if you added them up on a numbers basis. And so this is quite an interesting concept, and it's a really interesting space that's emerging that could be very relevant to diseases like these chronic ones I'm going to talk about. So just to give you a summary of the microbiome in the gut, this is the area where we know most about. So the gut bacteria do a whole bunch of things, and the gut is very important, say, protection against pathogens. The bacteria in the bowel will synthesize vitamins, including, you could add vitamin B3 to this, which is a really important one, potentially in motor neuron disease, with the data coming out there. This is where the immune system develops with the interaction between the bacteria and the bowel. And there's also various other factors that go on. So the key ones probably are the dietary fiber producing short-chain fatty acids, which are involved in this brain-gut axis, which again, we don't really understand, but we know that the brain and the gut can work in combination with each other to work through various processes. So all of these things have to function in our systems to produce good health. And I put this in because it's an interesting slide. I know we age ourselves over time, but the microbiome also ages. So when we're born, we start with no bacteria in our guts, And depending upon how we're born, whether it's a vaginal delivery or C-section, then different bacteria are introduced. But by the time we get to 3 years old, we probably have our adult microbiome in our gut. And that is sustained through life until we reach about the age of 60, where things decline. And they may decline differently in males and females with the menopause. Hormones are really important in this process as well. And so are different factors that come into it: diet, BMI, how fast the gut moves. All these things can influence the microbiome. And I'm going to talk a bit about this during my talk in the context of chronic conditions. And the other thing to mention, which is really fascinates me, is that in breast milk, there are oligosaccharides that we can't use as humans. They seem to be primarily there just for the bacteria, just to generate the right bacteria from the beginning, which we need to function. And so in the chronic disease space, there's some interesting things to consider. So many studies over the history of research really look at associations. And so things like Alzheimer's disease, there's an association with the pathology, Parkinson's disease, some pathology. But it's very hard to establish causality in humans. We can do it in animal systems if we know what the underlying drivers are, but in humans, it is an issue. And particularly in diseases like ME/CFS, where studies are small, and it's very difficult to do what we need to do, which are these longitudinal studies where we try and link and correlate changes in symptoms with maybe a biomarker that we measure in blood or an imaging system in the brain. So cross-sectional studies are the norm, and they cause problems, and we need to move beyond them. We can generate interesting data from a cross-sectional study, but establishing what is important, and you'll see this in my talk with these new techniques where we can find thousands of things that are different, but which are the key ones that we need to work on moving forwards that could allow us to develop new treatments and targets for that. And so I thought I'd throw this slide in because Brent Vaughan gave a great talk in the previous series about Alzheimer's disease and how he was using factors that could modulate the brain that could actually impact on these pathologies that we see. But most of the research has been around trying to clear these pathologies from the brain, either with a drug or a therapy. And we're actually hitting this late target really in the disease when the patients have dementia and Alzheimer's disease. But we know very well that changes occur before then, but we don't really understand how they fit in to the, so really, we really want to be targeting things much more early on. And maybe it's too late to target something in the brain, but if we knew what was going on here in these early parts of disease, then we may have more of an impact. And mitochondria do fail in Alzheimer's disease, as they do in many conditions. And so intervening here, but then how do we know who to intervene with? And this is the challenge really, who's going to be the ones that are susceptible to develop Alzheimer's disease? And so in terms of a chronic disease, and I'm going to come with a few examples here. So an individual will have a chronic disease. There was probably some cause back in time, and we don't quite know what that cause is in many conditions. There will be a trigger that would drive the patient to develop the disease, and there'll be some sort of driver that is going on now that maintains the individual in that state. So if you take long COVID, for example, we know that COVID-19 seems to be one of the triggers that triggered off these conditions, but not everybody gets long COVID. So it's probably much more complicated than that. And so there probably are other factors, other susceptibility factors that are important for somebody to develop long COVID that we have to really try and establish what they are. And again, there's a disease driver that's driving these conditions. So some people with long COVID with sort of post-viral short-term condition will improve, whereas others will develop a more long-term condition. So why? Why are people different? So what is driving that illness? And I thought I'd put this slide in. When I talk about ME/CFS or myalgic encephalomyelitis chronic fatigue syndrome, people always ask me, well, why is it called that? And there's an interesting history around this. So myalgia essentially means pain, and this encephalomyelitis is inflammation of the brain, spinal cord. But really in ME patients today, the ones that are diagnosed, we don't really see much of this. It's very hard to pick it up with standard techniques. Melvin Ramsey was the first person really to identify ME, and he called it ME back in the 1950s. There was an outbreak at the Royal Free. Over 292 staff were affected. Very few patients, interestingly, were affected, and over 262 were admitted to hospital. And based on that outbreak, he wrote a book which has been updated from that, and I'll come back to this in a second. So in terms of what we call it, it's a real political hot potato in this area. So the patients want it to be called ME/CFS because it seems more real than chronic fatigue, because chronic fatigue has been considered to be more of a psychiatric condition, which is a label that they really don't like. Whereas psychiatrists in papers will call it CFS-ME, which upsets the ME patients. And some people just call it CFS, which sort of inflames both camps. And I just sort of put this slide in. I'm not going to go through it, but there's an interesting MEpedia if you're interested in this royal free outbreak. So they followed up these people 65 years later, because it was in the 1950s. So one group of patients from that outbreak recovered pretty much completely, some nearly completely that had some sort of relapses occasionally if they pushed themselves. And there was a third group that really didn't recover at all, and they remain incapacitated. Some had paralysis. So it does look like, if you read the information on this in the papers, that this was a viral driver. There was a virus that drove this condition. But this led to quite a lot of interesting research in this area. Melvin Ramsey was one of the key people that tried to make people believe that this was not a psychiatric condition, which has been labeled, even this outbreak was labeled as a psychiatric condition, and these people were hysterical and making it up, and it wasn't real. And that's been a real stigma that's dogged this condition for a while. So this is a slide that shows a number of things. So there's similar graphics I showed you before. But if you look at the right-hand side over here, what I've done is I've put in some of the key symptoms that we find in ME/CFS. So on this picture, you have a healthy control group, which is the left-hand side. And then 3 different severity classes of ME. So the severe ones are over here. And this is the multiple sclerosis group, which we've been using as a comparator. And it's really useful when you're comparing patients with a condition with controls to also use another condition that is very similar. You can see there's very similar symptoms in the MS cohort, but there seem to be 2 quite distinct conditions. And as you can see, it's become more severe. So the patients over this side are very severe, and the symptoms are much more exaggerated. So if you look at ME/CFS as a disease into this sort of criteria, what were the drivers? And many patients will complain of a viral trigger, or maybe stress in some individuals, and that they can see that as something that happened in their past that they think may have led to their condition. But there's clearly other factors involved. So many of us had EBV as a child. I did, and I don't have ME/CFS. But why do some people develop the condition? And so again, there's something going on that's driving the condition. And what's potentially hopeful in terms of ME/CFS is that patients can vary. Sometimes they can be really sick and not so bad. So they're clearly fluctuating, which does suggest that we could find out what's going on. We could really intervene with the illness. And so this is an interesting slide, and it sort of highlights some of the problems with this condition in getting it recognized, not just by the medical profession, but society in general. And so you can see at the top of this figure, there's Jennifer Bray, who's a patient with ME. She became quite famous, produced a really good documentary. If you want to find out more about the condition, I'll suggest it's on Netflix. You can watch it. It's called Unrest. And she's given TED Talks. And then this is a family, the Strong family. Leaky Strong is the mum. Jamie works in my group. He's quite severely ill. He's moderately affected, but he fights it every day. And Laura, his sister, is housebound and can't get out, but they look completely normal. And when I give talks to ME groups, you look in the audience and you think, well, they look okay. But what they will tell you is that they had to prepare to come to that seminar probably a week before, doing very little, and they will suffer for maybe a week to 2 weeks afterwards from attending. So these people are invisible really by society. You wouldn't know that they were affected. And then you've got the other extreme. So Whitney Defoe he's got a famous father, Ron Davis from Stanford, who was very big in the genetics arena, in the Human Genome Project, and he is really sick. You can tell just by this. And so we have this sort of people that are forgotten, left at home, because really the healthcare system can't really help people like Whitney Defoe. And then there's other ones here that they look okay. And again, the medical profession in the UK or across the world cannot really do much to help them. And so what are the problems with this illness, and why has it been so difficult to get a handle on? It's really hard to diagnose patients. You probably see from the MS histogram at the beginning that the symptoms, there's a lot of overlap. So it's like a process of elimination, but because you're comparing to other chronic conditions, you really never get there's no test, there's no diagnostic test that you can give to a patient that will say you have ME/CFS. So that is a challenge. And also it's a challenge in terms of if you want to develop treatment. So there's a couple of treatments involved in exercise that have been used up until very recently. And all of the outcome measures are questionnaires or fatigue scores. And so there's no sort of hard data that you can really base whether someone's improved on. And that's been a real problem. And there's just huge distrust between the patient groups and the medical profession, mainly because the patients really feel ignored and treated as there's some mental problem there that they're dealing with. And so this causes big issues. And the key one really is there's very little research funding. It's not and also it's not an area that as a clinician you'd want to get involved in. So many clinicians steer clear of it, and they're warned off working in this area because basically there isn't really, it's not believed by everybody, and also it's very hard to see how you can progress. But it is an interesting disease to work on as a scientist, and I really enjoy what we're doing. I think we can make some progress here. So what are the main symptoms? And this has been another challenge. The symptoms have been really variable across the world. Different people using different criteria. But in 2015, the IOM report out in the US put together. So these are the 5 main symptoms of ME/CFS. So reduced ability to do your normal activities. And these can be very, very different for individuals. So some patients can't get out of bed. Some can maybe have a shower once a week. Some can get dressed sometimes. And some can maybe hold down a part-time job. So there's a huge spectrum of impairment here. They're normally tired, and that will get worse if they do more. Post-exertional malaise is a really important symptom. It's the one thing that seems to distinguish this condition from, say, people with depression, but it's very hard to quantify because it's really about whether the symptoms worsen after exercise or cognitive or emotional effort, and how do you measure that? And it's not easy. They really don't sleep very well, and that's a common feature of many other conditions. Cognitive impairment, they complain of what they call brain fog, where like a really bad hangover, I suppose, in terms of what they tell me, but the brain can't really function, they can't think, and it's very difficult to do things beyond maybe a couple of minutes, and it will get worse. The harder they try, the worse it gets. And this final one is orthostatic intolerance. There is a problem with the vascular system in these individuals, so when they basically lie down, the symptoms are much better than when they stand up. And so that's why many patients lie down, because that's when their symptoms are at their least. So these are the 5 things, but as you may remember from the previous slide, there are many other things that can crop up in this condition. I thought I'd put this in 2 other diseases which are very similar. So long COVID, I'll mention a bit more about that in a minute. So a viral trigger, as I said, they have fatigue, malaise after exercise, so very similar on many levels with ME/CFS. Shortness of breath is a little bit different, so maybe that's a very early form of ME or post-viral disease. Chronic Lyme is an area that's really neglected as well. Between 5 and 20% of patients who get bitten by a tick and know they had a bite and have treatment with antibiotics will develop chronic Lyme. And once you develop it, it is really hard to get on top of it. And many patients don't know they were bitten by a tick, so they don't know that they have the spirochete infection. And so those patients will suddenly come down with something with all the symptoms, but they've got no recollection of a tick bite. So it's very hard again to diagnose it when you don't have the original evidence that they were bitten by a tick. So we're very interested in comparing those 2. This is an interesting slide. It just compares many conditions with ME/CFS. So why are we studying this condition? And so the orange and the green line look at the mental component. So how bad is it to have this on a mental level? How bad is it to have on a physical level? And in the studies that have come out, it's one of the worst things to have. So worse than cancer, worse than diabetes, heart disease. And it's really debilitating on multiple levels. So it's really something we need to take seriously. And in the UK, there's a lot of people affected. So 20 million predicted worldwide, but I think the pandemic, unfortunately, will increase this number, with some people that have long COVID maybe slipping into the ME group and not really improving. Cost of the UK is staggering, but it's not just the cost in money, it's the cost in terms of looking after people with it, the parents, maybe the partners. They need some of these patients need full-time care. And many patients don't get a diagnosis, so they have to live with something. They could have this or may not. This is interesting. So over 70% of the patients are female, which is very common for these autoimmune type diseases, if ME/CFS is one of those. And this is demographic of when people are affected. So not only does it affect women, as you can see in the red bars here, there seems to be 2 peaks. So there's one in the teens. So children can get this condition, so they're off school, will cause lots of problems, they can tend to do better in terms of the long-term outcome than, say, somebody that's in their 30s that gets it at that age. Then they don't tend to make such a good recovery. But there's two peaks. The question is why? What's the difference? Why are we getting one in the child range and why one more adult? And men don't get it quite so much, as I said, which is quite common. So there's something interesting about males and females that makes the women more susceptible. This is an interesting graph from came out of Esther Crawley's group in Bristol. So there was a research project in the 1970s called the ALSPAC study, where they took, I think, around 40,000 babies at birth. And the idea was they'd study them over their whole lives. And so we got lots of exciting data on that group, who develops a condition at what, and you can track back. So this is a study of the adult group. In the paper, they also look at the children, and the children are very similar to this. And so what we've got here is the star is showing you when they were diagnosed, finally diagnosed to the point we could diagnose it. But if you look back at the number of consultations per year, they were going higher than the controls for 15 years before. Tests per patient go up, ramping up over time. But again, so this was developing for a long, long time before the final diagnosis. And this is the thing that we need to try and improve. If we could diagnose early, could we do more? Could we do more to the patients? Prescriptions, referrals. And so you can see there was a history of the patient developing the disease before they were finally diagnosed. And so is there a trigger point? Maybe before this, maybe before 15 years. But with long COVID, we have quite a defined trigger point that we can potentially try and work with these individuals to try and work out whether we can do something to stop them going into this long-term condition. Glandular fever, this is an interesting graph here. So you can see there was an infection with Epstein-Barr. 90% of people will have it for about a month. Some are seriously ill, others aren't, but the bulk of them will recover. So the poor 99% will enter this post-viral phase where they're still quite sick, and very few, 1%, will go into this chronic fatigue ME phase over here. And this is looking very similar to long COVID, and the numbers are remarkably the same. So about 10% of patients will develop long COVID. Hopefully most of those will recover, but we don't really know how many are going to fit into this group, because there's obviously a huge number of individuals that had COVID in the world. So this is where we are from a research point of view. It's been quite a lot of work in ME/CFS, even though it's been a challenge because of the funding situation. And these are the various groups that seem to be coming out as hard data. And the ones in the red rings are the areas that we're working on. I'll cover some of those today. But what we really need to do now is to try and piece together this puzzle. How does it all fit together? And can we start to do that? So I'm going to talk a bit now about altered energy metabolism in ME/CFS. It's an area that's been studied in some detail. Been studied on a number of levels. One is the metabolomics, looking at the plasma to give us clues as to what's going on systemically with the patients, with the muscle, the brain, and other organs. And this is the first paper that came from the Norwegian group, looking at metabolomics or profiling of plasma. And what they found was that there was much lower levels of amino acids in the blood of these individuals. And they speculated in the paper that this could mean that maybe there was a problem with the fuel use. You had to use more amino acids to make energy. There was an interesting component in this paper that looked at RNA profiling in cells from the patients, in peripheral blood mononuclear cells, and found some problems with the PDH system. And that system is the feeder, if you like, from glycolysis from the cytoplasm into the mitochondria. That's never been followed up, which it should be. And the other thing that they did was they took the serum from the patients and they added them to primary myoblasts, human myoblasts in the lab, and they showed an increase in mitochondrial respiration. So there was some factor in the serum that could impact on metabolism and potentially drive the cells to use mitochondria. And so this paper came out in 2016. There's been a few other studies. So the amino acid situation is pretty hard now. We really do see this in individuals. But we've been running some studies in similar vein to these ones where you add the serum, so we add plasma. So we started off with human myoblasts, and this is an assay that we developed in the lab. So we measure intracellular oxygen levels. So the cells in a dish will take the oxygen levels down inside the cell below the environment. And so we can measure that level, and what we can measure is when that changes. So if you look over here, cells on their own, when these cells are at 8%, they're running at 6% inside the cells. And if we add the serum from the controls and the ME, they both go down, but the ME patients go down further, meaning that the respiration has increased in the cells, which fits quite nicely with this paper. We've subsequently looked at different cell models, and we don't always get consistent results. And so we're following up on this, but it may not be as clear cut as there's one factor in the serum or plasma that can drive a change in energetics. There's things in serum and plasma that could probably also do that, even in a controlled individual. So that's an interesting bit of our work. In terms of what's been published, so big proteomic study by a group in New Zealand showing that there was problems with mitochondrial dysfunction based on what they saw at the proteome level. And other papers coming out of Newcastle University in the UK, looking at PBMCs. And I'll just talk a bit more about these two here. So this was a study from Australia looking at PBMCs from patients and what was deficient. And this one from Newcastle looking at skeletal muscle from patients, myoblast cultures, put into culture in the lab, and then looking at their biology. And so this complex five papers, I call it, for Miscellaneous. So if you try to measure respiration, oxygen consumption using something like a seahorse system that some of you may be aware of, they're very difficult to work with. So, you know, this is the levels, very low. You get basically, you can't really see much of a change in the system. So what they did was they transformed these cells with EBV, which is an interesting choice of virus to transform them. And when you look at the respiration, it rockets with the EBV transformation, but you don't really see much of a difference in total oxygen consumption between the patients and controls. But what you do see is some big differences in the stoichiometry of the mitochondrial proteins. So complex 5 and complex 1 were quite different in the ME group. So much more of these proteins in the ME group, in ME group cells. So there was a disbalance in the proteins, but even though respiration was the same in terms of output. So that was quite interesting. It's a transformed line, so we have to be cautious, but it was interesting that you could do that and see this in the lab. And then the group from Newcastle took primary myoblasts from ME patients, and they looked at their glucose-driven respiration, and they compared that to what the cells could do with either galactose or glutamine. So what they found, which is interesting, was the glucose respiration was lower, and these are stem cells taken from a patient, put into culture, and then looked at their function. So there was a difference, even at the stem cell level, when you developed a line from them. Whereas the amino acid fuel use was okay, which fits a little bit with the Norwegian group, suggesting that maybe there's a problem with glucose metabolism. So what this shows really, particularly this paper, is that you can actually transfer ME cells, patient cells, into culture and have a defect that you can see in the lab. And so what is driving that? Is it some sort of genetic or epigenetic, or what it also may be? There may be something that you're also putting into culture that's come from the system. So that's where we are really with the ME. So there is good data on mitochondrial dysfunction, but it's all around these blood cells. We haven't really looked at muscle, brain, or anything on a sort of MRI type level. And that's where we are. And so one of the things that we've been doing is to try and use this system called Metabolomics. And so you've probably heard of these other components here. So there's genomics, transcriptomics. These are true omics in terms that we can look at everything in a sample. So just to give you an example here, so you can sequence blood from a patient and look at the genetic profile of the individual. If there's any pathogens in the blood, say phage or bacteria, or other forms of yeast, we can detect those. So you can get a really good picture of what's going on. And the same for these other platforms. You can get a really good coverage of what's going on. But for metabolomics, even though we're looking at the output of the whole pathways, which you think would be really valuable, and it could be in the future, we can only detect a small number of compounds. So that's called targeted. So we can see compounds that we know what they are, but the bulk of the things we find in a human plasma sample are unknowns, untargeted. So we can see molecules, but we don't know what they are. But we can actually see what they do if we compare two cohorts with each other. And so what we're trying to do with our metabolomics work is to try and assess, can we get a handle of what's going on in the body, what's going on in the brain, the muscle, and other organs, by measuring what's in the plasma, so what's floating around in the blood. So we're looking at outputs of pathways. So this is a cell. You've got lots of biology, biochemistry going on in there. And we're trying to see whether these are ABCs, these are the metabolites that are generated by a cell. If they go into the blood, can they give us a clue as to what's going on in the body? Now, we don't know whereabouts in the body, but it's something where we can build up to see what's going on. So that's the cell level. But also we've got other factors that could be playing a role. So somebody could have a virus, or they could have a fungal infection or a bacterial infection, or there could be the bacteria in the bowel. And also there's lots of interest around toxic chemicals. What would they do to this process? Or even drugs. So someone's on medications as we get older. Many of us are on lots of drugs as we age. So all these factors can have a confounding effect on what we see. And so the idea is that we try and use this profiling of plasma to see what's going on in all these organs. And so what we're looking for is there a specific ME factor that could give us a clue as to what pathways are upset in these patients? Because at the moment, if you do a simple blood test for ME, there's nothing comes back as different. So standard NHS blood test, or in any country, you don't really find anything that you can nail it down. Say this patient's got this, is upregulated or downregulated and have this condition, or something's wrong here. But as well as the bacteria and the other pathogens, there's all these other factors that we have to worry about when we do this type of thing. So the age of the patient, what's the body mass index? And if somebody's really sick, so a sick patient that's bedbound, can have a real impact on their activity. And all of these things have to be taken into account. So it is a complex system, but the hope is if we look at enough individuals and enough samples, we could maybe pull out factors that aren't skewed by other things. So, you know, the yellow dots here can come through. So we've been doing this, and we've run a couple of studies. This is probably the best study. I won't go into detail, but you have to be really careful where the samples come from. This is something we've learned the hard way. This is a Polish cohort from our collaborators over there. Reasonably large study for ME/CFS, 54 patients, 23 controls. And what we're showing here, these are volcano plots. And so what we've done is we've taken the plasma, we've extracted it in different ways to sort of bias towards amino acids, lipids, and we also do the TCA cycle. And what this shows is that anything that's in the middle and the black is not significantly different, and anything in the pink is significantly different. So things in the top right or top left of these graphs are really different from the control group. And so if we use a cutoff of a 50% up or down, we're finding enormous numbers of metabolites that are different, over 1,000. So the problem we have now is to which ones are important, in the disease. And let's just give you an example of something that we're really excited about, but it's really challenging to take this forward. So this A9.65 over here in the red box, these are the levels of that metabolite. We have to account for drift on the columns. This is QCs across the run, so there's a bit of drift, but it's not too bad. These are the healthy controls. They've got a value of around 400. These are our patients, 40,000. So the question is, what is this? Is it a drug, or is it something really important? But not all the patients have it, but we currently can't identify it. So it's a problem to try and compare runs without knowing what things are. And this is something that we're trying to address by getting more funding. But unfortunately, the charities that fund this work, they're not really keen to fund something that is unknown, but we need to find out what's important. And this is things that we do know. So this is very similar to the first paper I showed you, Metabolomics study. So amino acids are really quite low. Glutamine's down by half. That's a huge difference. So all these are quite significant. And there's things here that feed into different pathways, energy pathways, either ketogenic substrate or TCA cycle. This paper is well worth a look. It's just come out by the Norwegian group, and there's a lot of the same lines as this. And so glutamine is low, so it's one of the most abundant amino acids in blood. It's down by half in a lot of the patients. So the question is, what does that mean? Glutamine mainly comes from muscle, but it can feed either into lipids, it can make glycogen, or it can be used in energy production. So we're not quite sure what's going on. And also the fact that it is low could mean that we're just not making enough. It may not mean that it's been broken down to use as a fuel. But the evidence is supporting that there could be a problem with glucose metabolism, and that's borne out. I haven't got time to discuss that too much today, but there is lots more evidence looking at high levels of lactate with exercise in patients compared to control. So there is something going wrong there. And in long COVID, we don't really have as much information to do in ME. I thought I'd put this into an interesting slide. So in acute infection, there's a whole bunch of things going on with inflammation. But Risa Pistorius and Douglas Kell from the UK have put forward this idea that there is a problem with these microclots in long COVID that don't go away, and they may be responsible for clogging up blood vessels, small blood vessels, and causing some sort of hypoxic event that is sort of transient in patients, and that could be a key driver in mitochondrial dysfunction, if you like, but more of a hypoxia. And there are various clinical studies trying to break these down with platelet acting drugs that could reduce this. And some of the evidence suggests that you may be able to, early on in long COVID, reduce the chance to develop a more severe condition like ME by breaking up these clots and going to normal. So I think watch that space. There's more money going into long COVID. There's quite a lot of MRI studies going on in long COVID at the moment. So there may be some more clues we can get from that will help in these other conditions. So I want to move on now, really, because many of you on this call are really aware of the sort of energy producing properties of mitochondria. But what about other roles? And one of the ones that I think is really important is their role in infection. And this slide just shows you, this is a slide of just mitochondria from a neuronal cell line staying. So with JC1, you can see the membrane potential of the mitochondria here. I want to draw your attention to these things over here. So these are what's called L-form bacteria. Now, most bacteria, or many bacteria, in either gram-positive or negative, can form these bacteria. So if we can see them in nature. If you treat a bacteria with antibiotics, you can drive them into this state. So in a wall-less state, they can't be hit by things like penicillin. And what one of our collaborators has found is that they're finding high levels of these L-form organisms, these wall-less organisms inside blood cells, white blood cells, predominantly from patients with this chronic condition. This is an ME/CFS patient. You can see the little green speckles here, and that's an L-form bacteria. And they basically just sit there at this point. They're not really doing a great deal. When they're in this form, they're very refractile to the immune system. We can't see them. There isn't a big immune response, and so they could be persisting in our tissues and bodies, and we really wouldn't be able to pick them up very well with standard tests. They can grow them in culture, and they form these really interesting structures. So there's no wall now, so all the sort of control of the shape has gone, and they produce these little blebs that you can see, and they just shed all of these into the culture medium. So you can imagine this happening inside a patient in the blood. If these were floating around in the blood, they would shed these fairly non-immunogenic factors that could do a whole host of things to the system. And even in a cell, what do they do inside a cell? So I'll come back to these in a second. And I'll just sort of show you this. So I've worked on mitochondria for a long time. And one of the things that I haven't really thought about in much detail before is what they look like in culture. So on this slide, you've got a typical electron micrograph, probably from liver from back in the 70s, where you can see the sort of classical structure of mitochondria. When we look with a light microscope, we can't get this definition, but we do see some interesting things. So these are human fibroblasts, which we work quite a lot with, and they're very long and thin in a human fibroblast culture. Very different from what we see in hepatocytes over here. So these are human hepatocytes we've grown cultured from a tumor resection and grown them in a culture, and very, very punctate, very much like this, but these are very different. And in one cells, I was going to show you a movie on this, but I probably won't, but this is a cell line where we put a GFP tag into the mitochondria, and then we've looked at what's active in orange. But you can see here cells are clearly not active, and if I ran the movie, you could see there's very different cell types. So even in a homogeneous cell population in culture, that the mitochondria are doing different things in different cells at any one time. So this is an interesting slide, and this is really the way we think about mitochondria in the immune system. So if you have a PAMP, or a pathogen associated collection of pattern, if you have a bacteria that's inside an individual, in a patient, you can trigger this innate immune pathway over here. My mouse has gone a bit crazy. I'm just going to move things. It's better. Yeah. So that's right. So basically various things from a bacteria will trigger what's more an innate immune response. So neutrophil activation, chemotaxis, those sort of things. And then if you have failing mitochondria, if you go through mitochondrial dysfunction, then you can see something very similar as an output. So the same processes are going on at the end, but different compounds and components modulate that process. And this is how we really think about mitochondria at the moment. And so this is really where I wanted to go with this. And so in 2018, Lida Piernas's group in cell metabolism published a paper that showed that mitochondria can be a defense system against pathogens. So Toxoplasma gondii is a very abundant bacteria, it's found in over 1/3 of humans. And what the mitochondria seem to do, they seem to surround the bacteria and engulf it. And by doing that, they then starve those bacteria of nutrients, and which is all very well. I mean, as you read the 2018 paper, there's a way of controlling the proliferation of bacteria by doing this. And there's probably some other mechanism in the cell that will get rid of the bacteria. But what this bacteria can actually do, then that can override this. So this Toxoplasma gondii can form tiny outgrowths. Here you can see in green, there's a little sort of vesicle here where it's upsetting the membrane of the mitochondria and breaking up this structure. So not only do we engulf them in bacteria, but the pathogens try and override that. So this is a good example of how we might deal with pathogens in inside systems. So this is a sort of a hypothesis slide, really. So if we have mitochondria in a cell, and each mitochondria can do every single function, then you may have, so this mitochondria here can make cofactors, it can make ATP, it can do reductive carboxylation, and it deals with pathogens. But is that really likely? And so the other idea would be that maybe we have mitochondria that do things at specific times, and depending on the cell they're in. So there may be mitochondria that make ATP and cofactors, do reductive carboxylation. In a cancer cell, there's probably more that are involved in reductive carboxylation, and some deal with pathogens. And those pathogen-dealing mitochondria probably rely on how many pathogens are in the cell. So if you have a healthy muscle cell from an active person, a lot of mitochondria make energy, and maybe there might be a little bit of infection in there or pathogen. So a little bit of the resources are targeted to deal with that and to wrap up the pathogens. But if you have a pathogen compromised cell, then it could look very different. So again, you have more mitochondria tied up with this sort of engulfment of pathogens to try and get rid of them, and less mitochondria available to make energy and other factors. And so this is the idea that I had around that. And so one of the things that I mentioned earlier was this post-exertional malaise, and it's really quite classic for ME and long COVID. So if a patient does more than their activity envelope, and what I haven't mentioned is every patient will have a threshold that they know that if they go above that threshold, they will suffer. But they won't suffer straight away, and they will tend to suffer about 3 days after the exertion. It can be the brain, it can be the muscle. And there's just a couple of images here. We were telling people until very recently to do exercise as a way of treating these conditions, and many patients got worse. So NICE pulled this treatment back in October last year, and so this is now not used. And so it's just a good example of the patient saying that don't use this exercise, it can make people worse. So why does this make people worse? Why do they crash? And how does this fit into that model that I just mentioned? So it's just another hypothesis, something we could easily test in vitro. So you're on the left-hand side, you have a cell from maybe an ME patient that has an infection that's compromised. It could be a virus, it could be bacteria. And so there's lots of mitochondria dealing with the pathogens in green. So suboptimal mitochondrial function, but they can survive and they have a certain level of energy. They do exercise and they push themselves. So they need more energy to do that. And so what may happen is that you then take away the control components, and more mitochondria are switched over to make energy, which allows the pathogens to get out of control and escape, and they will proliferate in that cell, may even go back to the sort of normal form if they're an L form. And what happens at 72 hours, maybe what we're seeing with this crash that we see is a rebalancing of the system. So the mitochondria have to get the pathogens back under control, which they do, but as a consequence of that, we lose the ones that were involved in energy. And so you're at a point below where you started. So this is just an idea, but it does fit quite nicely with some of the concepts that we see in the patients. And so that's really around about how mitochondria might deal with pathogens. And I want to come on to this idea of how we may make ATP outside mitochondria in an oxidative way in some tissues. Maybe we can think, or you can think about how this might fit in with what I've just discussed around dealing with pathogens. And so I used to work with a guy called Ian Craig, who was a professor in biochemistry. He was one of the first people that found mutations that could link to resistance. And he said, when I told him this would be considered treason by some. And this has been a problem for the people that have put this idea across for the last 15, 20 years, that nobody really wants to believe that ATP could be made outside mitochondria by some oxidative process. But I'm going to show you some evidence that maybe it can. And so this is a really important slide. So if you look at the middle part of the slide, if you look at the density of mitochondria in different tissues, they vary hugely. So the heart has more mitochondria than other tissues per surface area. Liver's not too bad, muscle reasonably high, but some are really deficient. So the brain has got about 5% surface area of mitochondria, and things like plasmablasts, shown over here, have very few mitochondria for surface area. Both of these tissues, particularly the brain, are really energetic tissues that use oxygen to drive their ATP production. And if that's true, how does the brain make enough energy with only 5% of surface area? Are they more efficient than other systems? Or is there another way that the brain is making energy? And the plasmablast is a really interesting model system that we plan to study. So this paper in 2018, so these are B cells. So when you differentiate a B cell, you go through to the plasmablast, which is the high energy antibody producing cells. So at this point, these cells have really cranked up all of their TCA cycle enzymes, all of the RNA to make respiratory chain proteins, but the mitochondria change very little. But what does happen is that the ER rockets enormously. So lots of protein synthesis going on, but there really isn't very many mitochondria. And so what we're saying is that this aerobic ATP synthesis is not happening in the mitochondria in these cells, but it's happening in these other structures, the ER in this case, and in the brain, perhaps somewhere quite different. So this is a really good system to study this. So these are rod cells from the retina in the back of the eye, the ones involved in dark vision. And so they're really, really energetic cells. And they have this really interesting structure. So you've got the nucleus here and the end plate, synapse, and all the mitochondria that you can see with a light microscope are in this inner segment here. So this one in the middle. So packed full of mitochondria. The real business part of the cell that needs the energy is the membranes, the rhodopsin membranes, where all the light harvesting pigments going on here. There's no mitochondria here. So that the general thinking is that the ATP diffuses up and there's enough of it to get here. So is that true? Is there really enough to make this much ATP per second when these cells are active? And what an Italian group did back in 2008 is they took these membranes from the rod cell of the eye, and they did a proteomics analysis of the system, and they found it was crammed full of mitochondrial proteins, ATP synthase, cytochrome C, not all mitochondrial proteins. You don't find TOM20, and there's a recent study by an Oxford group that's found exactly the same. So these rod cell membranes are crammed full of mitochondrial proteins, but I think the general theory thinking in the world has been this is an artifact and not real. And so this is an area that we're quite keen to look at, and we just started looking at this. This paper came out 2021 by the same group, Alessandro Morelli, Isabel Panfoli's group. And this is a really I think it's a really good bit of data that supports it. So what they've done here, they've taken mitochondria from the brain of I think these were bovine mitochondria. They've isolated myelin from the brain, and they've also taken these rod outer segments that I showed you on the previous slide. And what you've got here is the ability of these fractions to make ATP per milligram of protein. So mitochondria can make ATP, we know that. And what's interesting is there an interesting effect of ATP added to the external system. You actually quench the ability of those cells to make ATP in this system. In isolated myelin and the rod outer segments, you don't have the same inhibitions, suggesting they're quite different in what's making the ATP. There's more ATP made by isolated myelin compared to mitochondria, but there's an enormous amount of ATP made by these rod outer segments. This is really quite interesting, and you don't see this ATP effect if I've not mentioned that. So this was quite interesting for me to thinking there really was something here we should maybe look at in more detail. So I had a really good technician a few years ago that just retired, and she loves doing histology, and so we wanted to look at the eye. So we worked on mouse, which is smaller than the bovine system. I can't get over how complicated the eye is every time I see this and think, so how this is like a window into the brain, if you like, with all the neurons. So the cells that we're really interested in are here. So you've got the rod and the cone cells. So the rod cells are the ones that do the dark vision, and they're the ones with these different structures, the inner segment. And so what we've done is started to look at this. And so this is a rhodopsin stained rod cell here. So you've got the nuclear layer in blue. You can see that quite nicely. The inner segment where you don't get the rhodopsin, and then the rhodopsin's crammed into the outer segment of these cells. We've also staying with ATP synthase, shown in green. And we are starting to see some stuff. It's not really strong. I remember the talk by Doug Wallace about where ATP synthase probably is in the mitochondria, the tip of the cristae. So the structure of ATP synthase in a membrane system will be very different from what's in the mitochondria. So we may not see things in the same way, but I think there is something there. What's interesting about rhodopsins, so these rhodopsins here, in humans, they're not known to pump protons, but in bacteria, they really do. So you could have a proton pumping system in these outer membrane segments, which combined with ATP synthase could actually synthesize ATP in that system. And you can see this a bit better on this slide. So there is some stuff here, and we've got some more, it's more of a punctate appearance in these rods that we think probably could be real. But in our new projects, we plan to use some more sophisticated EM methods to look at this in more detail, and that's the grant application. We're just trying to generate some pilot data. This is the myelin sheath of the brain. So if there's not enough mitochondria in the brain, where do you make the ATP? And this is some ideas from Alessandro Morelli that maybe the myelin is the site of the machinery, the energy machinery that makes the energy in the brain. And there is some interesting correlates here. So this is a cyanobacteria. Again, a very membrane-like structure, no mitochondria, but all of the bits and pieces that make the energy are in these membranes. So this is very, very similar. There is literature that suggests that there are mitochondrial proteins in here, but it's all done a long, long time ago, and it's something that needs to be followed up on. So there could be mitochondrial proteins here. This paper came out very recently, which is really quite interesting. So these are oligodendrocytes, the cells that lay down the myelin. So tiny, tiny cells laying down meters of myelin in nerves. And they showed in this paper that there were components coming through from the oligodendrocyte through the myelin that produce factors that could regulate mitochondrial function. So it's not too far-fetched to think, well, maybe other things come through. Maybe respiratory chain proteins could come through into this membrane system. And just to go back to the pathogens, and so, okay, if we've got systems where we're lacking mitochondria, and mitochondria are really important in dealing with pathogens, how does the brain, for example, deal with pathogens if it doesn't have very many? And this may be the reason why we get these viral accumulations of viruses in the brain, lots of diseases that cause problems with the brain. We have a blood-brain barrier that can stop some getting in, but it could be a problem. So this is an example of what happens if you infect a cell with chlamydia. This Prosti's work from Germany. The mitochondria become very sort of network-like, like I showed you with the previous slide, the toxoplasma are gone. So the mitochondria sort of they engulf the pathogens, and maybe that's part of the process where they're kept under control. But if you have a virus infection in the cell, so you can see here, this is a reactivated herpes virus in the red cell here, you make the mitochondria become really punctate, so tiny, tiny puncta. And so they're going to be very different in how they function with ATP, and also how they function maybe with dealing with other things. This cell here is not reactivated. You can see the normal structure of mitochondria. And so the question is, what if you get both of these in the same system? And we're seeing with COVID and the pandemic, there's evidence of bacteria getting into the patient's gut, into the body. And so you may have co-infections that could be really relevant. And if they're both together, then it could impact on the mitochondrial response. And so these are the tissues. The heart seems to have lots of mitochondria. Maybe they deal better with pathogens than, say, the brain with very, 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 very few. And so it's an interesting concept, something we should think about perhaps moving forwards. And so I mentioned that with ME/CFS, there really isn't any pathology. There's no evidence of full-blown infection. So what's the evidence that there could be an infection going on in something like ME/CFS, chronic fatigue syndrome? And if you think about the microbiome, so the microbiome, as I mentioned at the beginning, so lots of work around the gut, but we're now seeing that we are not sterile. And so there are bacteria in a lot of our tissues, in blood, at low levels, but they're there. Lots on the skin, lots under the skin. And so there's this whole emerging idea that we could be doing things with these microbes. What are they doing? In the lung, there's over 600 species found in the respiratory system. What are they doing? Are they doing nothing, or are they doing something useful? But the gut is the area that we really know most about. And we know that in many conditions that if you go to a dysbiotic state, you tend to have a disease. So cancer patients, people with ME/CFS, other conditions, Crohn's, will all have a dysbiotic bowel. And what we try and do is to take them back, and you can do that in some individuals and see what happens. And I'll come onto some data on that in a minute. So this is a really interesting study. So as I mentioned before, the ME patients, the stigma of being labeled as somebody with a mental illness has really dogged the whole area. And this meta-analysis shows that people with schizophrenia, depression, and chronic fatigue syndrome have evidence of a leaky gut. So zonulin 1 is a protein that links the gut, like the glue that links the gut together. There's a high level in the plasma, high levels of lipopolysaccharide, which is a component of bacteria, and all these other endotoxins and things that come from the liver that are generated in response to bacteria. And it's across the board. And so all of these more mental illness type conditions are showing evidence of pathogens coming through from the gut into the body. And I suppose the question is, what are they doing? Are they relevant? Are they important? But they're not. We're not seeing this so much in controls. So there's evidence of things coming in. This is an interesting paper in ME for Ian Lipkin's group. Again, no real sort of full-blown infection, but this molecule came up in their proteomic study, CAMPLL37. So it's an antimicrobial peptide. It was raised in the plasma, suggesting that there could be some low-grade infections. Also, this thing here, that there were sort of a clonal antigen-driven B cell event. So these B cells are the ones I showed you that make the antibodies. So maybe they're exposed to something fairly specific that's driving the infection. Interestingly, this CAMP in the 1970s, these antimicrobial peptides also impact on mitochondria, which could also be perhaps playing a part of the process. I'll mention this briefly. This is some work that Soft Cell Biologicals are based in Utah, St. George. So they've been working for some time on different chronic diseases, and what they've developed is a way of culturing these pathogens from blood. And you can see, I showed you this one before. There's another L-form culture where you get these blob type structures. And as I said, these are quite stealthy organisms. They're not really that visible to the immune system. And what they found was that in conditions like ME/CFS and fibromyalgia, it was much easier to culture these L-forms from individuals. And you could get more L-forms from an ME patient. And if you have ME and fibromyalgia, even more. So there was some correlation there with chronic conditions. Over 2,000 subjects, thousands of these things, and many have been sequenced now, and many of them have never been seen before by man. So this is a really emerging field of science that we really don't understand. If you look at the cancer literature, papers coming out, there are cells seen around tumors that contain intracellular bacteria. Are they L-forms? And we haven't really got a handle on how to measure them yet. Something that we might do with our new work. So I think there's emerging, and this is a video, but I'm not going to show it. But one of the things that Soft Cell did, so Brent was the founder, sadly he died during the first pandemic, not from COVID, but it was very, very sad. But he was a plasterer by training, didn't go to medical school, but he had lots of joint problems, and he decided to look at blood. And this is a blood smear, and I can't show you the video, but basically in medicine, most clinicians would just look at a dry blood smear. You would not look at a live culture. So what Brent did was he looked at blood, and he looked at it over time. And you can see in the middle of this slide, there is a thing, I don't know what it is. It's like full of lots of tiny packets, and on the video it will pulsate. And over time in culture, you would see this rise. And the interesting thing about these patients with chronic conditions, that they pick these up time and time again. It wasn't just a one-off. It wasn't something coming from the outside that was growing in culture. And the red cells are also interesting in terms of they can produce sort of tiny, tiny aggregates, a bit like the long COVID story with the clots. So this is an area that needs to be studied in more detail. But I thought I'd mention this. So there is good evidence that these things exist in patients. And this has just come out last week. And this is a paper showing in seriously ill chronic fatigue patients, there is high levels of antibody against bacteria flagellins. Interesting, a bacteria that we find in the gut that's quite relevant. So I think there's an emerging space here about low-level infections in these conditions, which is something we need to get a handle on, and could be the way into treatment if we can find out what they are. And so in terms of what we're doing in my group, I thought I'd just put this up and say a bit more. We're very interested in these areas. So we think gut dysbiosis is really important in chronic fatigue, and I showed you that early study where they looked at the different schizophrenia, the depressed patients. So we think these pathogens are important, and they may come in through the gut route. How the energetic dysfunction fits in, and there's lots of theories you could put in around that. So this is the area we're working on. These other things on the other side are areas that we don't really have the capacity to do, but they need to come into play somehow. And I thought I'd just show you this as I finish off really. So in terms of how might the gut be involved in these conditions? And how might these, so there's some trigger, there's some sort of trigger in these conditions. And that trigger seems to lead to gut dysbiosis. We see this in long COVID. People with COVID infection, severe patients had a really quite nasty gut dysbiosis. And what does that do? It will allow the gut to leak. And that will leak into the bloodstream. And the question is, what happens next when pathogens enter the bloodstream? And most of us will deal with it with the immune system. So the immune system will deal with it. But for some individuals, maybe the ones that are susceptible, the immune system won't cope very well. And they could end up, as I mentioned, as these persistent organisms inside cells, maybe in key organs and tissues, not systemic, but they may be in joints, in the brain, places where they can hang out. And the big question is, how do the mitochondria become involved in this? Are they involved in some way? And is it the inability of mitochondria to deal with these pathogens that's important, that makes people susceptible? Or are there antimicrobial peptides that affect some people's mitochondria and others? And this is where we are with that. So last really on the sort of clinical side, I'm not quite sure I am for time, but I'm probably going on a bit too long. But if the gut plays a role in these chronic conditions, then if we improve the gut, it should improve the symptoms. And this is some work that we've been involved with a guy called Julian Kenyon, who Jeffrey introduced me probably to about 8 or 9 years ago at one of his meetings in London. And we've helped Julian with some of his studies. So what Julian does in a private clinic is he'll send off stool samples from the patient to a company called Atlas, and you come back with a report which breaks down what's going on in the bowel. So you've got the diversity of the microbiome, probiotics, beneficial bacteria, dietary fiber, and what Julian will use these three or four things is to give a clue as to what the readout of the patient is. So have they got a dysbiotic bowel, or is that something that he should maybe think about treating? So we've also been looking at a lot of his samples recently, looking at the raw data, and there's some interesting stuff coming out of these quite simple reports that you can do fairly cheaply. So what Julian did during the pandemic, he was using fecal transplant, so delivering donor stool samples to patients and showing some benefits, but in a private clinic, there isn't proper controlled trials. But what he switched to this different approach, because during the pandemic, you couldn't do fecal microbiome transplantation because of COVID, it was in the bowel. So what he did was he used what's called gut floral replacement therapy. So took probiotics, took out the bacteria and introduced them into the lower bowel, along with fiber and butyrate, which is one of these short chain fatty acids, and produced some quite interesting results. So this is one of his papers. It's not peer reviewed as far as I know, but in the journal there was some interesting changes. So these individuals, this is their profiles before treatment with the GFTR here, you can see, and there were some changes going across in different, and the outputs are fairly good, so clinical response. But really at the moment we don't have any good markers of what's going on in these individuals in terms of, are they really better, and will they relapse back to where they were. And so before the pandemic, I got these two together, so Soft Cell and Julian, and said that why don't you look with the Soft Cell approach to look at what's in the blood. Can you culture L forms, and can you sequence them? Can you find out what's in the blood, and does it change when you do a transplant, fecal transplant? And 10 subjects went over where we have baseline, treatment, and after treatment and follow-up, but only one's been looked at. I thought I'd show you it because it's quite interesting. So this is a slide showing the types of organisms found in the blood. So these are sequenced by deep sequencing from a patient that had a really nasty viral encephalomyelitis, really severe chronic fatigue, had seizures, quite unwell. So a real viral infection of the brain that caused something like ME/CFS. And if you look over here, you have two colors. So the pre ones are the, sorry, I'm just going to stop a second. Sorry about that. The sun's coming through the window and it's sort of blinding me, so I can't see the screen very well. Okay, so the orange are the pre levels, so number of species detected in the blood, and the post are after the treatment, in this case, the microbiome transplantation. So you can see the numbers, the total numbers gone down. And then if you look at the demographic of the types of organisms, there's a real difference. So the orange ones are before treatment. So Acinetobacter very high, go down to almost nothing. This one's here, Sphingomonas. A lot of these things that they're detecting are opportunistic. pathogens. You wouldn't expect to find them in an individual's blood. The grey ones are after treatment, and there's a big difference. So these ones here, the Pelamonas and these are normally found in healthy individuals. So you've got a predominance of these. So this is really interesting, and we're hope to do some more sequencing on these samples. What's also interesting was the what changes with symptoms. The DePaul questionnaire is a good measure of how fatigued somebody is and how bad the symptoms. So before treatment, they scored really highly and very badly. They were really quite severe. Straight after the transplant, it's a drop down, and 5 months post, they were really low. And as far as I'm aware, this patient is still well 3 years on. That's only one, but I think it shows that the power of potentially looking at this approach, running proper trials to really see whether this can work. So sequencing is something that you can do now on blood fairly cheaply. There's different levels, and it's something that we will do going forwards. So I'm going to come on to the last part of the talk, really, is around the Raman. Hopefully we've got time. And so how do you study these pathogens in cells, in systems in the lab, in vitro, and their interaction with mitochondria? And how do you study mitochondria? And so we had an interesting project with Spine UK. It's a 6-month pilot for the pandemic, but we spread it out during the pandemic. And so it's working with a group in engineering, Zhou and Wei Huang, and they use Raman spectroscopy. They're based in engineering over here. And we had Hariba, were a collaborator, whereas we didn't unfortunately work with them because we couldn't really, they've got some nice bits of kit. So Sarah was the postdoc on the project. And so, as I said, I'd really like to be able to see what's going on with mitochondria in cells. How could I tell a mitochondria that's making energy? How could I tell a mitochondria that's maybe making building blocks? And a pathogen, if I infect a cell with a pathogen, is there a clear difference in that mitochondria in a cell using a light microscope type system? And so the group in Begbrook in Oxford have been using Raman spectroscopy. We've worked with them for some time. So essentially you shine a laser at a system, and then we look at the vibrational energy of that material. I'll come back to that in a second. So this is the setup at Begbrook. It's quite an old system. And you see a cell down the microscope, and you can interrogate what's going on in that cell, looking at its readout. And we work with Hariba. Nathan was the postdoc in Hariba. It's only a pilot, so we had some input from them at the beginning, and we hope to work with them in the future. And so what is Raman? Essentially, materials can have a Raman energy. So basically you hit with a laser, bonds, chemical bonds, things like benzene rings will have a Raman scatter, and we can detect that with the Raman. Things like fluorescence can be a pain because it's quite a strong signal. But anyway, we're looking for Raman scattering. And so what you get if you look at biological material is a fingerprint like this. And so you can see some things come up, like glycogen's got quite a strong profile. Phenylalanine's another one that comes up quite nicely, lipids, but it's more of an overview of what's in the sample. And so we've been using this technique for quite a while in our ME work to see whether we could develop a diagnostic. And I just mentioned that briefly before I talk about the mitochondrial work. And this is some work that we've done initially, looking at Raman as a way of examining cellular metabolism. So the top panel shows some standard assays. So these are cancer cells, 143Bs. We've known for a while now that what drives these cells to use glycolysis in culture is the media of glucose concentration. So 25 millimolar glucose will do very little oxphos, and you can change that by making them more physiological. Probably one is more tissue, so they will respire. You can measure ATP, and we can see here that you can see the transition, so probably more glycolytic here. They go down, they use more mitochondria, as you can see with the respiration, so the ATP. But what was interesting is when we ran the Raman spectra of these cells, we saw some quite different profiles. So this bottom profile has got high glucose, and there was a huge difference here in this peak that's potentially really important. And these blue arrows are the cytochromes. You can pick up cytochromes really quite well with Raman. So this suggested to us that maybe we could study mitochondria using this system, but this is a whole cell. And so what we did during the pandemic is we worked with different cell lines. So these are 2 cancer cell lines that are quite different in their how they make energy. So this one here, A549, is mainly glycolytic in high glucose, but we know that if we treat it, run it in 1 millimolar for a couple of days, it will switch. It will switch to oxphos. So we have an oxphos phenotype. This one here is very oxphos, but it doesn't really care what glucose it's on. It will do it on both concentrations of glucose. So this is what we see down the microscope. This is a mitochondria, and then we can see a profile. If you look at other things, a peroxisome, or they'll look really different. So you've got a very clear readout of what a mitochondria is under a microscope. So what we did in this experiment I'm going to talk about is we took the two cell lines, we made mitochondria, and the cells were grown in high glucose. So in high glucose, we'd expect the A549s to not use mitochondria as much, and the A460 would. So these are isolated mitochondria. And what you can see here is that the A549s are really not doing very much at all. When we add substrates or uncoupler, we don't really see much of a change, but you can see quite nicely here with the H460. If you just put them in the system, we can see respiration. And if we add the fuel, the succinate, they go high, and if we uncouple. So what we did was we took these mitochondria at these early time points when we knew what they were doing, and then we looked at them with the microscope. And so this is an example of the outputs. So from a different experiment. So this is the H460, 1 millimolar glucose, really cranked up respiration. And you can see here on succinate, and when we add succinate plus ADP, FCCP, and antimycin A. And there's some clear differences on these profiles. I'm not going to go into too much detail, so it could take half an hour. But you can see here when we add ADP, just the succinate, there's a big change in this peak. So are these making ATP? And is that something we're measuring? That's very close to the cytochrome peak. And here's a big transition here. And when you uncouple or poison, you do see big changes in the profile. So we're quite excited about this as a way to move forwards. The other thing we can do at the single cell level is to look at the impact on metabolism. So using those profiles that I showed you of the Raman spectra, this is a study using different drugs. So we've got metformin, which is a known inhibitor of complex 1, nicotinamide, which is something people have been using as a potential anti-aging molecule, and tanganol is basically an amino acid. And you can separate these out really quite nicely. You can see here no drug, metformin, different concentrations. And so not only can we see a difference in the patients, we can also see a difference with drugs, which could be useful going forward in working out whether there's changes in metabolism. So you could imagine taking these cells and doing other things to them, like metabolomics. So where do we go next with Raman? This is my last slide, really. So I think we've studied mitochondria in isolation, and we're just about to study them in cells. And we think we can maybe look at the different pathways. And how do we study these L forms in vitro and in patient cells? And so this is going to be a challenge. There's no stain that will stain them, but if they've got a unique Raman signature, then we may be able to pick them up, not only in culture, but also in cells from the patients. And I'll stop there. Probably talked enough. So this is just an acknowledgement slide. So my group with the people in red have done a lot of the work I've talked about today. There's Janet who did the microscopy, and the various collaborators and funders on the bottom. So thank you for listening.